One can see the streets without trees having the temperature of 50 degrees, and where there are trees, it is the temperature is just half of that. So one should promote the plantation of the trees to make your environment cool and clean. Now, the arrangements of the bacterial cells. Bacteria show characteristic bacterial arrangement of the grouping, and that's why they are that pattern of the division is just going to recognize them that these are of which class of the bacteria. These arrangements are due to the pattern of the division attachment of the bacteria to the mother at the time of the cell division. I am going to tell you why there are the pair bacteria, why there are the chains. Means binary fission occurs and the daughter cells remain attached even after the division. This arrangement occurs in the cocci, bacilli mainly, but it can be also in the spirochetes, which is not that important, which is mostly in cocci and bacilli. So the cocci arrangement, when these are multiplying, the cocci may form pairs, chains, grape-like structure, tetrads, and the cubical packard, and that is the arrangement diplococci. Diplococci arrange in pairs means cocci divide in one plane and remain together. As one can see, these are the meningococcal bacteria and the gonococci. Diplo means two in pair. The pairs have pointed ends. Streptococcus pneumonia in pairs with the kidney bean shape. These are the Neisseria bacteria. So the pairs with a bit different pointing ends and the shapes, they can be seen as the different bacteria. The bacteria can be in chains. Cells adhere after repeated division in one plane, and streptococcus means chain. So these are found in the chains. These include streptococci, pyogenes, enterococcus, lactococcus. These are in the chains. This chain could be short or it could be long. Here one can see the single cocci and streptococci. These are in the chains. Diplococci, as one can see, these are in the pairs. And sarcina, when they are in the groups of 8, and the tetrads when they are in a group of 4, and the staphylococcus when they are in the grape-like structure and they are adhering to one another in a clump of bacteria. So one can see here the pairs, and these pairs are seen in case of meningococcus and gonococcus. And these structure of the bacteria is because of the division of the bacteria. If you just look at this, one bacteria, when it is going to divide, it is going to become diplococci, and the bacteria are joined together one another. So, and when they are in the form of the chain, these are called streptococcus. And here one can see the bacteria which are having the division in 4 planes. These are in the 2 planes, X and Y. These are the tetrococci. Similarly, those who are in the 3 planes, X, Y, and Z, these are the sarcina, and those which just divide in every plane, these are in the form of the grapes, or they are in the form of clumps, which is called staphylococcus. Now, arrangement of the bacterial cells. These are the grape-like structures. I have told you these do divide in every plane. So the cocci in the irregular groups or the grape-like structure, because the cocci divide in the random planes, not in the proper planes. So these are just grape-like. Tetrad, group of 4 means square group of four cells. Here, cocci divide in two planes like micrococcus, sometimes called tetracoccus because of their two-plane division. Cubical packets, these are groups of the eight cells because divide in three planes, and these are genus Sarcina, sometimes called octacoccus because of eight bacterial combination. Gram reaction, Staphylococcus and Streptococcus are gram positive, whereas the diplococci can be gram positive or gram negative depending upon the retaining of the stain they do have. So the coccus is single-celled. Diplococcus, these occurs in pairs. Tetrad, group of four cocci. Sarcina, cube-like shape. Streptococcus, these are in the chain. And the Streptococcus, these are in the form of clusters and group. So wrong is wrong even if everyone is doing it. You never follow the crowd because the crowd do settle somewhere. Right is right even if one is doing it. So try to follow or go alone because you can reach to some destination ultimately. Arrangement of the bacteria, bacilli arrangement, bacilli split only across their short axis, so form limited pattern, not as in case of the cocci. When multiplying, bacteria rods separate, not remain attached to one another. They do form pairs sometimes, so-called diplobacilli, as in case of the Klebsiella bacteria. They do form chain, so-called streptobacilli, as in case of Streptobacillus species, Bacillus species. They form branching chains, as in case of Lactobacilli, and palisade arrangement, as these bacteria are joined together longitudinally to form a hedge, as in case of the plants. So this is the arrangement which is called palisade arrangement. One can see the coccobacilli look like coccus and bacilli, bacillus, two pairs, this is the palisade hedge-like. Streptococcus, these are in the form of chains. Then there are certain bacteria with different shapes, while in the division that can be club rod or enlarged rod. Here one can see the single bacillus, the diplobacillus, the two joined together, streptobacillus. These are just having in a line, and then the palisade, one can see how they appear on the microscope. Then bacilli arrangement, these are can be massed together like Macrobacterium leprae. These are just piling upon one another to make a heap of bacteria. So that is called massed together. Then cuneiform or Chinese letter, they remain attached at the different angles, various angles to each other, and do not leave their parents, and they resemble the letter V or the Chinese letter. These are the characteristic of Corynebacterium diphtheriae. These can be recognized just at the microscope by having this particular appearance. So here bacteria divide by the binary fission, and daughter cells remain attached to the mother cells. Even after the division, they do not detach and get separate to remain attached to give the appearance of V-shaped ball. Here you can see the mass together as in case of the TB bacteria, and here you can see the Chinese letter or cuneiform bacteria. These are attached to their mother. Now one can have the shape of the rod ends by which a very skillful and very sharp microbiologist can recognize that what these bacteria are. Flat or the square ends, they stick like brick-like. These are in case of bacillus. Rounded end are of the rods are in case of salmonella. Swollen ends, the rods, club shape, these are in case of the Corynebacterium. Tapered cigar-shaped rods, these are the fusiform, as in case of fus- Fusobacterium. Bifurcated rods of the Bacillus and the Clostridium do form resistant spores in unfavorable condition. So, gram reaction, gram negative rods are group of the Enterobacteriaceae. Gram positive rods include Clostridium species, Corynebacterium species, Bacillus anthracis, and Listeria monocytogenes. Here one can see that different shapes of the bacteria, Coccobacillus, palisade, chain shape, pear shape, and these are having spores in the bacteria in the center of that, and there have the spores at the one end. Then similarly the rod-shaped bacteria and the other shaped bacteria. Now coming to the size of the bacteria, these are, as I have told you already, these are usually of 0.5 to 10 micrometer long. Unit of the measurement in bacteriology is micron or micrometer, that is 1,000 of the millimeter, and it can be seen by the light microscope. Smallest bacteria are the mycoplasma, their size is 0.2 micrometer, chlamydia 0.3 micrometer, rickettsia 0.5 micrometer in diameter. These are equal to the size of the largest viruses, pox viruses, which are having about 0.3 micrometer in size. Smallest organism which can exist outside the host independently, these are the smallest bacteria. The longest bacterial rods, Borrelia, can have the size of some yeast and a human RBCs. The diameter of the smallest body that can be clearly with naked eye can be seen is the 200 micrometer size, because less than that is can only be seen microscopically. So the size matters. One can see the human cells and one can see the bacterial cells. So many bacterial cells can lodge inside the human cells, and this, as one can see, is the 10 micron size, and these bacteria cocci is 1 micron size, and the bacilli is a 2 micron size. So these are the measuring units. 1 meter is equal to 1000 millimeter. 1 millimeter is 1000 micrometer. 1 micrometer is 1000 nanometer. And similarly, the smallest unit that comes to the angstrom, which is the smallest unit of the size of the bacteria. Again, one can see the 1 centimeter, and then comes down to the smallest size, which can be seen only by the microscope. And here is the largest bacteria of different sizes. Then some of the bacteria are of RBC size. The past is where you learn the lesson. The future is where you apply the lesson which you have learned in the past.